Mendel was not satisfied with tracking just one trait, though. He also tracked the inheritance patterns of two different traits using dihybrid crosses. The goal of dihybrid crosses is to determine if the different traits, say seed color and seed shape, are inherited independent of each other or if they're inherited together. So let's look at the data for both seed color and seed shape. In this system, the yellow color is dominant over the green color, and the round shape is dominant over the wrinkled shape. So similar to the monohybrid crosses, the P generation were two individuals with different homozygous genotypes. One parent was homozygous dominant for seed color and homozygous dominant for seed shape. The other parent was homozygous recessive for seed color and homozygous recessive for seed shape. This means that the resulting F1 generation were heterozygous for both traits. And here we can see the big Y little y for seed color and the big R little r for seed shape. Now, when those F1 generations underwent a self-cross, we see that the results are pretty complicated. All the possible combinations of phenotypes are represented. Yellow round seeds, green round seeds, yellow wrinkled seeds, and green wrinkled seeds. And Mendel had consistent results with all the dihybrid crosses that he performed. The individuals that showed the dominant phenotype for both traits, such as yellow round seeds, were the most common. The individuals that showed one dominant trait and one recessive trait, for example, round green seeds and yellow wrinkled seeds, were equally as likely as each other, and the individuals that showed both recessive traits, green and wrinkled, were the most rare. And these results consistently showed a 9:3 to 3:1 ratio. This led Mendel to conclude that the alleles of different traits are inherited independently. And now we know this as Mendel's law of independent assortment. That is, alleles of different genes sort out independently of each other during the process of producing gametes. And we know that independent assortment occurs during metaphase 1 of meiosis when the homologous chromosomes line up along the equatorial plane of the cell.